Good evening to all of you, and very good morning to Dr. Suresh Kumar.
Good morning.
I welcome all of you, all the participants, on behalf of Regional Science Center, Tirupati. And you know very well, today is October 21st. Today we are starting our first Nobel lecture series. Fortunately, this day, October 21st, just now I was browsing and could see that this is the birth anniversary of the great Nobel founder, Sir Alfred Nobel also. So we are very fortunate to have this first lecture on this day, start this first lecture. And now I'm going to the speaker without taking much time. We are here having a senior scientist from the Jansson pharmaceutical, the companies of Jansson and Jansson, Pennsylvania, USA, Dr. Suresh Kumar Swaminathan with us. I hope all of you welcome him from your place. A brief introduction on Dr. Suresh Kumar here. In fact, we are very fortunate to have him with us in this evening. Dr. Suresh Kumar had his graduation from University of Madras with a gold medal and masters from Madurai Kamaraj University, and after staying probably few months, I don't know, maybe few years also, in JNCR, Jawaharlal Nehru Center for Advanced Research in Bangalore. He moved to Heidelberg, Germany, where he obtained his doctorate in life sciences from Ruprecht-Karls University of Heidelberg, Germany. And after that, he served as postdoctoral research associate and also research scientist at University of Minnesota in USA, and also served as serving as a visiting researcher at Mayo Clinic. His current research involves development of biologics of several disease indications, pharmacokinetics and molecular biology of Alzheimer's disease, amyloid beta peptides across the blood-brain barrier, bioanalysis and quantification of small molecules in biological matrices by LC/MS. He has designed and successfully developed nano drug delivery formulations for cancer therapy with over 10 years of productive research experience, exemplified by peer-reviewed publications and a patent which is also commercialized. Scientific expertise spans cancer biology, Alzheimer's disease, molecular and cell-based assays, expression screening of cDNA or small molecule libraries, studies with small animals. And once again, I welcome Dr. Suresh Kumar. Without taking much time from all of us, I request and invite Dr. Suresh Kumar to please take the stage and present his lecture. Thank you, Dr. Suresh Kumar. Welcome you.
Thank you. Thank you for that kind introduction and a long introduction. Very nice of you. So, am I audible to all of you? Yeah, okay.
It's clear.
So I see a very diverse audience here joining, and first of all, thank you all for taking time to join this evening. And also, once again, I thank for this opportunity and as well as for this kind introduction. So like the title of today's presentation is basically attack to the recent Nobel Prize, Nobel Prize ceremony, and this is for discovery of hepatitis C virus. And I just from the introduction people might have made. So what I would like to do today is that I would like to keep this presentation not as a monologue, but as a dialogue where possible. So please feel free to interrupt or ask questions, or if there are something that we need to discuss, we can pause and discuss on that. And second thing is that this lecture is intended for a very wide audience, and then who is not necessarily familiar with the field of virology or even biology, I would say. And as well as there is a need for I would say experts and professionals as well. So I would just like to join in as you guys feel where necessary. All right. Next slide, please. Okay, prior to going into the lecture, I just have some statements to make. So the contents of this lecture are pretty much on my own, and then this is based on whatever the data that's available in the public domain and in the public literature, especially from peer-reviewed journals and books or articles. And then the data that is being presented here is not generated or currently not related to my current role or my organization. So any liabilities, good or bad, all rest with me. And I don't have any other conflicts of interest to declare with respect to scientific data. I'm not here to hard sell anything. So I once again, I just want to keep this lecture more open and then invite the platform. Next slide, please. Okay, so I'll just start with the heroes for this evening, and then what you see on the screen here are three gentlemen on to the right side of the screen, on my screen at least, and these are the Nobel laureates who were awarded a Nobel Prize for Physiology or Medicine for 2020. And so they are Dr. Alter, Rice, and Houghton, and they are currently now at NIH at US or at Rockefeller University or at University of Alberta at Canada. And so on the left hand, what you see is that is a award or the prize. What you get if someone wins a Nobel Prize is that they get three things. One, definitely they get a Nobel medal. That is shown here, and then that medal has an image of Alfred Nobel, and then they get a diploma or a citation, just a paper certificate showing that they did in fact were awarded a Nobel Prize, and then they are Nobel laureate. And third thing that they get is the cash award. A typical award prize is 10 million Swedish kroner. That's approximately 8 crore Indian rupees. And so this is shared between three of them. So the award is pretty much is not really, is not known for the cash prize, or it's not the monetary reasons that it is known. It's more prestigious because it is still considered one of the top-notch award to get, back in the world. And most people in the scientific arena think that or endorse that this is by merit and merit alone. And so because of those aura attached with it, the Nobel Prize is the most coveted prize in the world. And of course, I'm really talking here. So the Nobel Prize is awarded for multiple disciplines. So the purpose of this talk covers the Nobel Prize in physiology or medicine. And next slide, please. So what we are going to do in next 40 minutes or 45 minutes is that just try to understand what did they really do to get this Nobel Prize, and then is that all the hype surrounding this and then why they were awarded is worth the merit or not, and is what I just want to really bring to you and your attention. And then in that course, like I have some discussion points and I also seek some inputs from you. Let's see how it goes. And then I have structured this lecture into three parts. First part is that I just want to really introduce to you this audience here to a general introduction to the world of viruses. Yeah, we are living, like a strange times. So viruses is in news, like you turn the newspaper media all across the globe, like one thing is common is a virus these days. So what are these viruses? Why are they really having this sudden attention? That something is what we just want to really understand here. And just a very simplistic understanding of virus. Viruses are very small particles, that's all. They are very small obligate or intracellular parasites. What does that mean is that they are small particles and then they require some host cell, some other cell for them to survive and reproduce. So in this context of definition of virus, so some biologists, some people think that viruses are not even a living, quote unquote, a living creature. And then, so this is always, some sort of interesting debate always goes whether viruses are considered living being or non-living being. But for that debate relies on where or how someone defines what life is. So for this purposes of lecture, what we would just consider is that they are very small particles. So when I say that they are very small particles, so how small are they? That's a question, right? So small can be small, very small, tiny, very tiny. So what are we meaning by small? So on the right hand side in the screen, you have a scale. That's a regular scale we are all used to. This is a regular centimeter scale. This is a linear scale, meaning that the units are in a linear fashion, increasing order. Scale here is not linear, it's on a logarithmic scale, meaning that the units, if you really see, they are jumping tenfold. 0.1, 1 nanometer, 10 nanometer, 100 nanometer. So if you really see this, what is indicated in the red box, the flu virus, is typically in 100 nanometer size, or the diameter of a flu virus is so small that it is not visible by eyes. We need some sophisticated equipment to visualize them. So those sort of objects or entities are called as microscopic or submicroscopic particles. And then, just take home, I would want to convey is that a virus is as size of 100 nanometer size, a typical size. It can range from 5 nanometer to all the way to some tobacco mosquito viruses can be around, very large than this. A typical flu virus is of a small entity. And now compare that to a chicken egg, like an ostrich egg, that is in the millimeter scale. Now, I'm asking the audience here, if you have a 100 nanometer virus, and then if we just ask, let's do a mind exercise, and then if we really start, ask to, pack this virus, make them stand one after the other all across the edge of the scale. So what happens? So this is 100 nanometer. So within this 1 centimeter distance, right, which is approximately 10,000 micrometer, we can make 1 lakh virus particles to stand back to back, shoulder to shoulder, along the edge. So in a typical 10 centimeter scale, what we can have is that 10 lakh virus particles can be made back to back. So the take home is that, like a small surface, anything that we touch, we come across, like the doorknob or anything like a mobile phone surface that I have, can have tons of viral particles just embedded on that surface. All right. Any questions there? Next slide, please. So what are these viruses? What do they do? I just said that, they are small creatures, let's call creatures or particles, something, it's just like a dirt or dust. So what do they do? Like we all get now, we are living in, like era of grocery shopping by, by post and then courier, and then we are used to getting packages. So viruses are just, like I would like this audience to visualize them as just packages, just like we get, like every day a courier. So a regular courier would have something like whatever, let's say if you ordered some sort of, like a ball, and then it's wrapped in some sort of, like bubble wrap, and then there is outer wrap or outer carton. So something very similar to this, a virus has 3 major components, I would say, is that they are packed, like they are just a packages. So the package has some sort of a outer material, just like a carton, and they are packaged containing genetic material. So genetic material is key for viruses to propagate, replicate, and divide. And so that the genetic material can be 2 types, as people know. So it can be DNA or RNA virus. Okay. And then, so on the left-hand side of the screen, what we have here is that, so if you really see that there are 2 strands, if you just see the 2 in green, so that is the genome or genetic material that is packed, and then surrounding that is a bubble wrap, it's called as capsid. And then often, like outer envelope or outer package is present, and sometimes this outer envelope may not be present. And then this outer envelope is just like a blue ring, and then this is just made up of fat and then proteins, and they are glycoproteins. And then you just see some protrusions, and those protrusions are mainly for going and attaching into the other surfaces. Okay, so this is a very neat, very well engineered structure they have. And so all that, so just for this take home, I would like to have from the slide is that they are just a package containing their small particles tightly packed of their own genetic material. It can be DNA or RNA. Next slide, please. Okay, so, like biologists always like to classify things. Like not because that they are not having any job or any other reason, but they want to have some sort of a structure. Yeah, so, so generally, how would you classify something? Like I don't want to really overwhelm you with all the technical terms. And then in a very simple classification is just by looking at how the virus or the properties of the virus. That's how biologists tend to classify broadly, like whether the genetic material, as I said, is made up of DNA or RNA. You can just, like these are 2 different classes of genetic material. And then the next category they would like to see is that whether this genetic material, what is the shape or structure of this genetic material. If you really look into the left-hand side, I have that the genetic material can be just like a single thread. They are called single-stranded DNA, or it can be as double threads. They are double-stranded DNA. And so, the next level of categorization is based on whether it is a single-stranded or double-stranded. Then, like people just, it's like scientists are products of the society. How like a common man would really categorize or organize stuff at home in kitchen, or like that's a typical way they would also perform in the lab. So they would just like go about how to categorize whether this a virus, when they observe under sophisticated instruments, whether it has envelope or not, whether there is a capsid structure or not. And these are some of the ways they really categorize. And then one important category comes by the of the host choice. Some viruses can like infect animal cells. Animals can be like our pets, like dogs, cats, and human beings. Some viruses can infect only plants. Like if you really see, I think when we get tomato or bindi, if you see that like those sort of black color mold-like stuff are basically due to the virus infection. And then there are tobacco mosaic viruses or bindi mosaic viruses. There are unique viruses that infect the plants. And then there are viruses that really infect bacteria. They exclusively just infect bacteria. And then last is that how they really divide. When the replication mean that replication is that they really divide. And then how they transmit, so that viruses can transmit by airborne or they through food or through blood. So the way they are transmitted. So it, like just depending on the properties, like biologists tend to really classify these viruses. And so on the right hand side, like what I have mentioned here as a Baltimore classification is the general way with which biologists approach this. I think it's like we don't need to really look into this more closely. Just this is for information purposes, that like scientifically people give this names like group 1 virus, group 2 virus, group 3, and so on and so forth. But what I just want to really convey to this audience is that not to get overwhelmed by all the scientific jargons. What essentially people are doing is that like they are categorizing these viruses based on these properties. Next slide, please. Like again, as I said, like when now you just have multiple ways with which you can really categorize the viruses. When you really combine, mix and match, you have different properties in different families, different sizes. And so, like there are multitudes or multiples of tons of viruses. And then we really, virologists and microbiologists have only studied very minuscule world of viruses. Like that's the take home I just want to really convey from this slide. And then I have really highlighted two like circles here or ellipses here in red. So I don't know if you're able to look or zoom into that. What that is essentially is that it's example for one is for hepatitis virus, and then the other one is for coronavirus. And they all belong to a category of what are called RNA viruses. What does that mean? Is that they all have RNA as their genetic material. In some sense, like I would just, like I apologize to the purists among the audience. In some sense, like hepatitis virus and coronaviruses can be considered as, like cousin brothers, but they are not one and the same. Like I don't want you to take and say that they are one and the same, but they belong to a same category of RNA viruses family. Next slide, please. Next slide, please. Yeah. Okay, I just want to really, like pause here and then, just put some questions for the audience, and then either you can really quickly, take turns to, answer, and just as a interaction or discussion. Like often in these times, we use the term, my Facebook post went viral. Okay, or the social media post, my Twitter trend went viral. What does it mean? Any guesses? When we say viral, what does it mean? No answers?
propagated large.
Yeah, it's propagated large number. Thank you. And it's just that, like it has multiple people have seen this, and then multiple people have really spread that message. Similar to what virus would do, virus can really propagate faster, or it can replicate, it can make copies of itself several times. So that's why it's called viral. So we have taken some of, so the next question is that I just want to really bring to this audience and ask the question, are all viruses are bad to humans?
No, maybe some viruses may be good, sir.
So which viruses?
E. coli.
E. coli. Okay. So I just want to really, um, um, like the take home for this audience, I would say, is that, like I don't want to categorize all viruses as bad. We are, like it's unfortunate, and then it is a great injustice we are doing to those small creatures, because when we talk of viruses, we always talk of viruses in the context of infection, like cold, flu, or coronaviruses, but then there is more to the viruses than they are causing infection. So just that, like um, viruses all, just the take home I want to have is that viruses are all not bad. And then just a point I just want to correct is that E. coli is not a virus, it's a bacteria. Okay. And then the next question that I want to really ask is that, like why is that some viruses really infect animal cells, some viruses infect bacteria, some viruses infect plants? Why is that? Any thoughts, students?
Maybe for reproduction.
Yeah, reproduction. But why not all viruses can target, let's say, eyes of a humans? Why is that each one is really, like infecting different hosts or going towards different hosts? Go ahead.
Maybe they depend on that particular thing, sir.
Yeah. So they
Survival.
depend on, for their survival. That is something is very important, is that they don't want to, like, I don't know how many of you have heard this proverb, don't put all your eggs in one basket. So it's something like this. It's an evolutionary mechanism. So viruses don't want to all, imagine all viruses go and infect only one organ, there is a stiff competition, right, to go and then use that as a host. So they just go and then seek new opportunities. So they just choose, try to mutate and then change themselves and then modify their behavior and then go into and then seek new woes. That's what happens. So it is pretty much for their survival. That's the reason that they have multiple O's, like they can pretty much infect different cells. That's very unique among the living creatures, okay. Last but not the least, should we eradicate all viruses in the world? Now we know, like we are talking about vaccines, therapies for, coronaviruses and that. So, like if we have a magic wand and then if we have to, should we ask for a wish to really eradicate all the viruses in the world? Is that going to be good or bad?
Bad, I think.
Why is that going to be bad? Okay, I think that's. I think I take that answer. It's not good to eradicate all the viruses in the world. So they are doing a very important place and role in the evolutionary chain. And then just imagine, so there are so many pathogenic bacteria, microbes in the sea, in the, bacteria infecting the plants. And so these viruses act as a check, to eradicate them. And if we really eliminate all the viruses, what happens is that their potential hosts can go really crazy. And then this will really alter the dynamics of the living world or the planet as we know. And so it's important to keep in mind that, eradicating all viruses is not a good thing. Okay, just want to go into in the interest of time, I would like to move into second part quick, and then next slide, please. In the first part, as I end, just to summarize what I want to really, take home for this audience. If one thing that you take home, at the end of 1 hour, so this is what I would like you to really take it away, is that viruses are parasites. They cannot survive on their own. They need a host cell for dividing, propagation, multiplication, however you call it. And then they are metabolically inert. That means they are just useless outside their host. They are just waiting for an opportunity or suitable host. And then the host can be anything, an animal cell, plant cell, or bacteria. And then there is a symbiotic there is the evolutionary relationship between the host cell and then the viruses. And then viruses are classified based on their properties, and then hepatitis C virus is an RNA virus, and so is coronavirus is also RNA virus. What does it mean? That means that they have RNA as a genetic material. And viruses can get transmitted by multiple routes. Again, wide multiple routes. It's also because of survival. So if just imagine if it is through single route of transmission, and if we really eradicate that route of transmission, they lose their survival advantage. So they just can adapt to the changing scenario, and then they can really get transmitted through air, blood, contaminated food, or water. And not all viruses are pathogenic to humans, and world without virus is no good. And then next section. Next slide, please. Okay, so I would just want to really go quick into this section. What's hepatitis? What is hepatitis? Hepatitis is commonly known to this audience as jaundice, as we call. And hepatitis is a very long history. It's closely associated with human history. This is a disease that we know for almost 5,000 years. And then this is a documented medical text available, or are describing hepatitis. So common symptoms of hepatitis. So on the right hand side you see a liver. Liver is organ for metabolism, for digestion, and then it plays a very important role in our body. And then what happens in hepatitis is that this liver gets damaged, and then this leads to abdominal pain. People get tired, jaundice, and then the skin and eyes become yellow. And in extreme scenario, the liver gets is not functioning properly, or very, very rarely it can lead to death. And as I said, we know this closely associated with human history, and then first documented cases were known even 5,000 years ago. And in a text, or in archaeological text, what they have really found is that it's very interesting that 5,000 years ago they thought a devil named Ahazu is infecting or affecting the liver. They don't know what the devil is, but they do figured out that this is a damage to the liver, even 5,000 years ago. But that devil, it took 5,000 years for humankind to identify and discover what the devil is. These are different hepatitis viruses. And then this is documented by Hippocrates. And then, as I said, world is not facing the pandemic like viral infection for the first time. Humankind has seen this viral infections, pandemic-like situation several times, and then just that we really don't realize. During Second World War, there were documented 16 million people, more died or infected by the hepatitis, or people had jaundice. And then, and it's also during the World War time, people really paid meticulous attention to how this jaundice spreads, and then they really put this jaundice into two bins. One bin is called as epidemic hepatitis or jaundice. That means it just spreads through food or water, contaminated food or water. And then another jaundice is blood-borne or serum hepatitis. And then they just categorized into two two categories. That sort of information for the first time, like historical distinction was made in 1950s. Okay, so although it is 5,000-year old disease, like we started to make some sort of a progress in understanding only in late 1950s. Next slide, please. And then, like I just want to fast forward, to 2000s, and then in this 50-year span, what has happened is that we really knew a lot about hepatitis, and this is a very devastating disease, and or the jaundice. Like the most common hepatitis or jaundice is of viral origin, and then this can be by different viruses. So as I call this, a virus that causes hepatitis or the jaundice is a hepatitis virus. And there are multiple types of viruses, hepatitis virus. And then the common is just like alphabet of hepatitis virus, hepatitis A, B, C, D, E, different categories of hepatitis virus causes this viral jaundice. And then there are some cases, very less common, non-viral origin of the hepatitis can also occur. This can be, like people who are commonly drinking alcohol, like they have liver damage in the long run. And that is because of, like it leads to cirrhosis, and then this will be a hepatitis. That's very less common. And then some rare metabolic diseases also results in the liver damage. But these are very minuscule portions, but large portion is driven by viral hepatitis or viral disease. Next slide, please. So I just want to really summarize here. There are, there are two major, although, like there are multiple viruses that can result in the hepatitis A, B, C, D, E. There are two major routes or two major virus classes that categories that we want to really focus on. One is hepatitis A, and then another bin is hepatitis B and C. Hepatitis A is commonly, like propagated or transmitted from contaminated food or water, and hepatitis B and C are through blood transfusion. And in 1950s, like early 1970s and 80s, like it's a big thing to have a very safe blood transfusion. And like we have made a significant progress in in having safe blood transfusions with our understanding of this viral hepatitis. And what you see here in the cartoon down is that this is a viral hepatitis. This is just not that, like someone gets jaundice or hepatitis C infection and then the disease comes the very next day. It's a very chronic disease, means it takes as long as 10 to 30 years for the disease to really make very severe. So if you see that, like in the cartoon there, from left to right, you start with a healthy liver, and then it is infected with the hepatitis virus, and then it becomes fibrous. It's called a cirrhosis as a condition. So, and then this might even increase the chances for liver cancer or carcinoma development. And then it's a long process, 10 to 30-year process. And oftentimes people, never even realize that they were infected with hepatitis C. That used to be the problem. And next slide, please. So someone had asked in the chat, what happens once the virus gets inside the body. I just hear, once the virus just gets in the body, it just goes to like it's just like if you see what happens. If we see our friends, we just go and start talking, right? They go to their friends or friendly cells in the body. Then first step is that they just attach onto the cell surface. What you see in the brown is a cell, and then they attach to the cell surface. And then once they attach, the cells just swallow it, and then they get internalized. And what happens is that then once they internalize, they really get they replicate. They use this cell as a factory to make more copies of themselves. Next slide, please. I'm not really going into the details here. I know it's all CDC guidelines are from, like on the different categories. This is for people who are more interested in the different the biology and then the epidemiology of hepatitis. So the main point I just want to really highlight is that two things. We really know quite a lot about this hepatitis virus. And second thing that I want to really highlight is that hepatitis A, again, is that gets transmitted from food, contaminated food, but hepatitis B is through some body fluids like saliva or things like that, and hepatitis C is mainly through the blood transfusion. That's pretty much the high-level summary of this slide. The next slide, please. Okay, so I just want to really pause, and then next couple of minutes, I just want to take these questions. Given that like hepatitis C virus gets transmitted through blood transfusions through blood, do you think that whether mosquito bites will transmit hepatitis? No, mosquito bites will not transmit hepatitis, thankfully. Like otherwise the world will be full of, like people infected with hepatitis. Okay, next question is that whether drinking boiled water will minimize the spread of hepatitis C, hepatitis A virus? Yes, as I said, like hepatitis A spreads through contaminated food and water. So when we really drink hot water, boiled water, so the virus gets killed. So drinking boiled water is recommended by the government agencies to minimize the hepatitis A spread. And then the next question is that will social distancing mitigate hepatitis C virus transmission? Pretty much no. The reason is that the social distancing work for coronaviruses. Why? Because coronaviruses are spread through airborne infection. Yeah. So whereas hepatitis virus is mainly through blood transfusion and then body fluids or like saliva or through the contaminated food, water. So even if we do social distancing, this is not going to really prevent the hepatitis C virus transmission. Can hepatitis reoccur to the same subject or same person can get 2 times hepatitis? It is very rare, but still it is possible, like very, very small cases. Hepatitis or jaundice can occur more than once. And especially if it is infected by different hepatitis. Someone can get first time hepatitis C born and sometimes it can be infected by hepatitis A. But, like those chances are, like it's extremely rare and unlucky if someone gets it. So I want to really close this section 2 and then just go to the next slide, please. All I want to really take home, without dwelling so much into the jargon, is that hepatitis virus C or it's something a virus can result in a very bad liver damage. And then this hepatitis infection can transmit through multiple ways by blood, body fluids, or contaminated food or water. That's all I just want to really, summarize from this section. Let's go into the section 3, which is really highlighting what this gentleman who did for the virus discovery did. Okay, so this is the problem, I'll just, we are living in 2020. We want to really go into world of 1960. What was happening is that, in 1960s, someone really got hospitalized, and going into hospital, getting blood transfusion is a big thing. It's just like people would think that they might not even come back surviving. Why? The reason is that often people really used to get hepatitis infection through the blood transfusion. They do know that. Okay. And people, what really puzzled was that in 1967, a scientist named Bloomberg, he identified the hepatitis B virus. As you say, hepatitis B virus, we just saw, it spreads through body fluids. So they thought, in 1967, scientists were really very happy. They thought that, they solved the problem of, hepatitis through the blood transfusion. They thought that, with the discovery of hepatitis B virus, they solved it. Okay. But what really happened is that in spite of screening for the hepatitis B viruses prior to the blood transfusion, people still got hepatitis infection. So it really puzzled them quite a bit. So they really did not understand what's going on. They initially thought, oh, so, we ruled out hepatitis A, which is like from contaminated food or water. We thought that hepatitis B is the major cause for the liver damage. And we now screen the blood for hepatitis B and then make sure by, multiple quality checks that the blood is clean. It does not have any hepatitis virus. But when we really give this to the patients, they still go and get the hepatitis C. Hepatitis C. So what is going on was really it's just mind-numbing for scientists in 1960s, and it was really a 30-year work from 1960s that scientists really tried to understand what was going on, and then they identified there is one more type of virus, hepatitis C, that is really the major driver for all this cause of unexplained hepatitis that were observed in the patient. And with the discovery of hepatitis C, and then we are able to, come up with diagnostic kits so that, now anyone who gets blood transfusion, they not only are screened for hepatitis B, they are screened for hepatitis C, and then the blood transfusions have become more safer. And in this way, we have mitigated to a large extent the hepatitis, viral hepatitis. And so this is the entire story in a nutshell of the discovery of this hepatitis C. And then just next slide, please. And then this puzzle, right, like solving this puzzle from 1960s, it took nearly 30 to 40 years for the scientists to really understand this in a greater detail. So what did these three people do? This is Harvy Alter, and he's at NIH. NIH is the primary research center at United States. It's National Institutes of Health. So he was the pioneer. He started this, his work was all done in 1960s or 70s. Imagine, he did this work in 1970s, and then Nobel Prize comes to him in 2020, nearly almost 40 years or 45 years later, then he published this work. What he did, he was trying to understand what is really going on, in this case of non-A, non-B hepatitis. So what does that mean? As I said previously, described to you in a very elaborate fashion, people got up when they get blood transfusion, and then they did get hepatitis, and then these blood transfusions were screened for viral hepatitis, so HCA or B categories, and then they were made sure that this is non-A, non-B type, and they still really went ahead, and then they got the jaundice. So what did Alter did? What he just basically did is that he really collected samples from the patients, especially the blood samples, and then he took that and then injected to chimpanzees, our close relative, and then he demonstrated by multiple experiments that this non-A, non-B hepatitis is also caused by another virus. That's all he demonstrated, but by painstaking, very painful experiments and using very careful studies, he just demonstrated this puzzle. When people really go, even if you have screened and ruled out A, B, so if people really get hepatitis, that hepatitis is also caused by another novel virus. So he was the first pioneer to really say that, this is also caused by a virus. And then, so he started, in a way, this experiment that was done almost 42 years ago was what now awarded a Nobel Prize now. So the take home here is that, at least in science, there is no, immediate successes. Sometimes the progresses are very incremental. So next slide, please. So the second person who got awarded Nobel Prize this year is Michael Houghton, and he is currently now at University of Alberta in Canada. But when this, during this early 80s, he was in a small company in California here in USA. And, what he did is that he used cutting edge molecular biology techniques, they pioneered those techniques, used those techniques for the first time, right? And then what he really did is that he identified, what that virus was. He did identify virus, and then he named that virus as hepatitis C. And then why, his contribution to this field is very enormous, because some of the techniques or the experiments I have listed here for somewhat more for professional audience here, this is very fascinating scientific discovery. That's how people name it. And then he finally said that whatever the average, the previous guy initially said that it is likely to be if someone gets a non-A, non-B hepatitis, it might be driven by virus, and then he confirmed that by demonstrating that is indeed by a virus, and then he named that virus hepatitis C. And he did not stop there. He further subsequently went and developed a blood test for diagnosis of hepatitis C virus. So almost all the blood tests now we have, all started or initiated with this preliminary blood test that was initially developed by Michael Houghton. And then just see here, this work was done in 1989, 1990. So this almost after 30 years, his contribution is now being acknowledged. And that's the next slide, please. Next slide, please. So this is Charles Rice. He is the third person. So he was really, asking a question, whether there was only one virus that is causing this infection or hepatitis, or there are multiple viruses. There is no evidence at that time that it could be only one single virus that is causing this disease. So he was at the University of Washington here in St. Louis in US. And what he did is that he really, did multiple experiments. He tried to infect chimpanzees with these viruses, but he failed multiple times. Then really, he went to the drawing boards and identified why these experiments were failing. And then he modified the genetic sequence or the RNA sequence of the virus, and then reinfected the chimpanzees, and then demonstrated that, it's not multiple, several types of viruses are causing this disease. There is one single virus type. It's unique, and it has a unique genome. It's hepatitis C. And then he demonstrated in 1997 that, this virus alone is sufficient to cause hepatitis C infection or liver damage. So in essence, if you really see, the story started in 1960s, then 70s, then, multiple experiments by 3 different groups at 3 different places. And then they just piggybacked on previous findings. And then what did they achieve over a period of 30 years is that they understood this, are contributed to our understanding of this deadly disease, and also made our blood transfusions much safer. Next slide, please. So this is essentially the summary of what these 3 people did. And this is from the Nobel Prize citation. What they got is that, you can read here, it's a 3 sequential steps. Harvy Alter was the first one who demonstrated that there is something unknown virus that is causing this hepatitis C virus infection. And then Michael Houghton and his lab and his team, they demonstrated that he named and he identified hepatitis C virus, and then he also identified kits for diagnosis of hepatitis C virus. And Charles Rice, who came last but not least, he said that, like this virus alone, he does not need any other friends. This virus alone is sufficient to cause this liver damage. And next slide, please. Okay, and like as we really talk about these three people who were pioneer and then won the Nobel Prize, there are two unsung heroes in this entire story. And this is Mr. Chu and Mr. Ku. And on to the left hand, what you see is Michael Houghton, who is the scientist who got awarded for Nobel Prize. But Mr. Chu and Mr. Ku were like working in the laboratory of Michael Houghton, but they were never awarded this prize for a very trivial reason that Nobel Prize could be shared only with 3 people, and the Nobel Committee does not allow prize to be shared for more than 3 people. But what is important is that all the methods, the blood transfusion kits, all the painstaking work, like were done by these 2 people, Chu and Ku. So they are considered in this field of virology as, like unsung heroes, and the latest edition of Nature has a huge cover story on them. So what's important is that, regardless of whether they are contributed and whether they go and get the prize or not, what's important is that their contribution, is helpful to the humanity and then for people all across the globe. We don't know who's Chu and Ku, and I'm pretty much sure this audience would never have heard this name until now. But we are all fortunate that we were, reaping the benefits of the pioneering work done 20 or 30 years ago by these people. So we should be enormously thankful to the hard work and pain and then labor they put into this and then develop these methods. The next slide, please. Okay, so why, something that you want to really chew upon and take home, why this prize is significant? As I said that, blood transfusions, current blood transfusion are much, much more safer than what was in standard of care in 1950s. And then not only that, now we have these discoveries, seminal discoveries that span 1970s to 1990, early 2000, were also very key in developing therapeutics for hepatitis C virus. We have now cure for hepatitis C virus. So we screen the blood, and in spite of it, someone goes and get, hepatitis C type. And then we have now drugs to completely cure or near complete cure for this virus. So this is the significance of this prize. The next slide, please. Okay, so I just want to, as I said, right, there is a cure that is available, and I am already at the bottom of our, just wrap it up in next few minutes. And what we really, what is the major challenge? The major challenge is that this virus, as they propagate, they really also mutate very fast. So that, that is one of the major challenges. And then we can use only chimpanzees to study these viruses. And then as we know that, for ethical reasons, chimpanzees are prohibited for medical research nowadays. So because of these challenges, our progress are developing novel therapies or cure with hepatitis C is showing low progress. In spite of this, there are multiple cure available, small molecules that are available now for hepatitis C virus. You have multiple small molecules, combination therapies that are doing remarkably well in the last 10 years. Next slide. Next slide, please. Okay, so this brings to a question: hepatitis C virus vaccines. Although we have therapies for hepatitis C, the vaccine is a very pain. And then the story we have not really made huge progress in developing vaccines against hepatitis C. As I said, the main reason is that this virus really propagates and then mutates so fast. And then, it's, it's much more smarter than the scientists all across the world. So it's very difficult to keep up with this virus. So, so there is and it's not unusual, there are other RNA viruses also for which, developing a vaccine is always a frustrating experience. One is HIV. We have cure against for HIV, but we don't have vaccines for HIV. So next slide, please. And so compare this with SARS, the entire discovery to Nobel Prize and then therapy and cure, it took nearly 40 years or even 50-year journey. This is a remarkable scientific success story. If you really talk to, our grandparents and ask how it is like to get a blood transfusion in those good old days, they will have all those scary stories. And they will make even people stay outside the house if you visit the hospital, because they thought that they will bring some sort of ill will or some infections from the hospital. Those are, days of the past. Now with SARS-CoV, what happened? From the discovery, from the initial pandemic identification at the source country, we have the entire sequence. We know the entire horoscope of this virus within 2 weeks' time. Right? So we have made significant progress in understanding this virus. And within months, we have really, multiple vaccine candidate trials across the globe by different governments and different scientific institutions are ongoing. And also there are very promising therapies, that are emerging for this COVID. And so what is that we should not really say, oh, that's a 50-year story. And then all those learning experiences have contributed to accelerate our pace of research and then keep up with these viruses. And what I want to communicate or summarize to this audience, we have been facing these viruses for last 5,000 years throughout our evolution. Okay, and certainly viral pandemics or endemics are not going to be the last. Okay, so but what is really important is that, next slide, please. Next slide. Is that what is really important is that how do we really keep up with these viruses, and then just how do we stay on top of this evolution game is the question. Okay, we have already surpassed the timing, and I just want to really read out these questions and then just put your imagination. So, what precaution one has to take to limit hepatitis C virus transmission?
Body fluid. Why?
To drink hot.
Hepatitis A.
The food is for A. So, whenever there is, going to a hospital, someone is going to have a blood transfusion, make sure, ask the doctor or nurse, is the screen for hepatitis? And then, minimize, limit body fluid, walking with the bare hands or touching saliva or other body fluids. That's one of the easiest way to limit the transmission of the hepatitis C virus. And can a patient get infected by both types or multiple types of hepatitis virus, hepatitis C and B? It's very rare, but it is still possible. And especially in some Middle Eastern countries, this sort of rare things do occur and, surprise the doctors. And especially where the food quality or food supply chains are impacted, and then the contamination of water supply is a problem. This can occur, but then when these dual infections occur, this is a huge problem because it leads to a severe liver damage, and curing is a major problem. Okay, last but not the least, whether the hepatitis C virus story is providing you hope that science can help overcome current pandemic, or it is just a story of Great. So I
Yes,
want,
sir. We
yeah,
hope.
yes, I want really, like the young, particularly the younger audience, to really be more creative, and then believe that, like have faith, or believe that, like we'll be able to overcome this pandemic and situation. It's not going to be easy, right? Like you're going to have newspaper coverage saying one vaccine failed, the other vaccine failed, this vaccine worked, that vaccine. Like those are all noise. But what is from our experience with this virus and other viruses, we can really take home is that, like we will be able to overcome. We have survived in this planet for last several thousand years by really waging this war against viruses, evolutionary war. So we would be able to overcome this as well. So I just want to really stop here with the message of hope that the viruses are not bad creatures, but we often do a disservice to them because we always relate to them in the context of disease. And the last slide, I have a take-home message for you. Just the last slide. So this is a quote by a famous biologist at Oxford University. What did he say? This is that the discovery and treatment of hepatitis C virus is a remarkable story, like 30 years ago to now. And then, like this is a fundamental success story of how a basic science, someone, when they really were just going and exploring and infecting these monkeys, they never knew that they are going to identify a cure, and then this will transform billions of people across the globe. But it started as a small experiment that really, like transformed the quality of life across the globe. But like the story does not end. It is not like a happy, like Bollywood movie ending. We have a lot of problems still, especially despite all the progress. Still, 71 million people across the globe are getting infected by hepatitis C virus every year. Like all the education, all the knowledge we have, this is pity. So I think it is now we need to really rely on our public health officials, like how to mitigate this. And then 4 lakh people die every year of hepatitis C virus across the globe, especially in middle income countries. This death rate is even higher. So something is that I just want to leave as a thought exercise, how smart, like we should be, like to prepare for a future pandemic, endemic, viral transmissions, how we should really, like we are seeing with all the progress, it takes at least 1 to 2 years to turn a discovery to a therapy or a vaccine. So how do we really curtail this time? Can we use emerging technologies like artificial intelligence and then really be smart at it? And then these are something that some young audience can really think about it. So this is something, like will be a primary ask from you guys when you really grow up, and then, like in your world, this will be a major question. All right. And with this, and I would like to conclude here. And if there are any other questions, I can take.
Thank you so much, Dr. Suresh Kumar. Do you have any questions, audience? Suresh Kumar can take 1 or 2 questions also from you. If you have any question, please unmute your sound and please ask the question. The Suresh Kumar's explanation was very good, actually, which is not even prompting any question to all of us. Okay, so nice of you. And now I request my colleague Sri Purushottam to please come over and offer formal vote of thanks to all our audience as well as to Dr. Suresh Kumar, please.
Thank you, sir. It's been a great evening for all of us. So once again, I wish good evening to all the participants. As we were discussing something about the virus, let me put the vote of thanks in the similar way. I hope all the participants have been contaminated with the knowledge or wisdom of science behind the hepatitis or jaundice, given by our speaker, Dr. Suresh Kumar. I feel it is a great tribute for this day, as it is the birthday of Alfred Nobel, and we have started a series of webinars related to the Nobel Awards or Nobel Prizes. And today we have started with the Nobel Prize in the field of physiology and or medicine. I'm sure that this webinar has many ways showcased the in and out of a jaundice or hepatitis virus. As we know, even for this day, there are many misconceptions related to the hepatitis and jaundice. I wish the participants, like the teachers or students who have participated today, carry this information to the society in eradicating such misconceptions associated with this hepatitis. On behalf of Regional Science Center and all the participants, we thank Dr. Suresh Swaminathan for accepting our request and enlightening our participants with the importance of the discovery that gained the Nobel Prize to the scientists. The same way, we should thank all the participants who have taken the time to attend this webinar, and we wish all the participants to join in similar webinar series which we will be conducting very shortly. And thank you for participating.
Thanks, and then have a rest. Good evening.
Thank you, sir.
Thanks for that. Bye-bye.
Thank you, sir, for this wonderful session.
Welcome.